paper that we have recently published. Got it. A paper that we have recently published, and because this is a growing line of research in my lab, I would love to hear your thoughts. All feedback are welcome. And also in the style of NYU, questions during the talk would be very welcome. If you do that, I would feel at home. Please don't hold back and ask any question that you may have. Okay, so the work that I'm going to tell you about today has been led by a brilliant former postdoc in my lab, Gokyo Okazawa, who is currently an assistant professor in the Institute of Neuroscience in Shanghai. It was also assisted by Christina Hatch at NYU and Alan Menku and Christian Makens at Champlimont. Okay, decision-making does not require an extended introduction for your group, but I would still take a few minutes just to familiarize everyone and remind everyone of the basics. Many of the decisions that we make require sensory inputs, and we need to take into account contextual and mnemonic information and other contingencies, for example, the time that we have for making the decision in order to render the final choices that we have. Many of these decisions are quite basic. For example, in a natural environment, animals would be looking for food. They would recognize objects around them. They would categorize them as food or non-food, and then they would take actions, for example, approaching them, grabbing them, and eating them. However, decision-making is not a reflexive type of behavior. It takes into account contingencies, and observing or finding food in the environment does not necessarily lead to an approach action. For example, if next to the food there is a dominant co-specific or an animal that could potentially be a predator, animals would probably not approach it, and they may decide to leave. So context is quite important for deciding what type of actions should be made. In order to be able to make flexible decisions, at least two processes need to be present in the brain. One of them is taking sensory information in and processing it to extract feature information. And the other one is using feature and object information in order to make the decision based on the context that we think we are in. This is a specific example that I gave you, which is making decisions based on images of objects, has also not been directly studied in the past, or at least not extensively. There is a long history of studying object processing in the ventral visual pathway, starting from V1 and going to intratemporal cortex. And we, for example, know that there are patches of cells in intratemporal cortex that are selective to faces, and that there are also cells selective to almost all sorts of visual stimuli that you can imagine. Decision-making research, especially perceptual decision-making research in the past, has largely focused on the frontal parietal cortex, and many of the examples of perceptual decisions that have been extensively studied have also relied on the dorsal visual pathway, not the ventral visual pathway, the classic example being the discrimination of motion and the line of work that was pioneered by Newsom, Mofshan, and Chadwick. Okay. We decided to ask how these different systems for object recognition and perceptual decision-making would interact with each other. And specifically, we decided to do that in the context of two tasks. One task is the standard direction discrimination. Another one is a face discrimination task that I will tell you about. Direction discrimination has been studied extensively in the past, both the sensory basis of it and the neural mechanisms for decision-making. The stimulus is a very simple stimulus, dots moving on the screen, and the percentage of dots that move coherently could be modulated to make the task a little bit more difficult or a little bit easier. And at the end of the stimulus, the subject, human or monkey, would make a saccadifying movement, or in some experiments, reaches to two targets, one indicating rightward motion, another one indicating leftward motion. And if the subject is correct, of course, it would be rewarded. And if the subject is wrong, it would usually not receive reward or be punished, for example, by a short time. Okay, as I mentioned, we can control the difficulty of this stimulus very tightly by changing the percentage of dots that move coherently in the same direction. If all the dots are moving leftward, it would be a strong leftward motion. If all the dots are moving rightward, it would be a very strong rightward motion. And in between, you can create a full continuum of left to right with different strengths. When subjects do this task, very nice psychometric function and also chronometric and confidence functions could be generated. Here I'm showing an example psychometric function where the probability of choosing the rightward target changes from 0 for a strong leftward stimuli to 100% for a strong rightward stimuli. I told you that this task has been extensively studied. I'm going to just highlight responses in 2 brain areas, MT, which is the direction selective region in the monkey brain. Neurons in this region are highly selective for direction of motion, but also for the strength of the stimulus. When you give the animal, you show the animal a weak rightward stimulus and record from the activity of rightward cells and leftward cells in MT, there is a small difference of activity and a slightly larger firing rate for the rightward pool of neurons. When the motion becomes more strongly rightward, the difference of the activity of the 2 pools of neurons would be clearer. And for the strongest stimulus, which is 99.9% coherence, fully coherent motion, the difference between the 2 pools of neurons is whopping. One pool of neurons is totally suppressed and another one is responding at hundreds of hertz. Okay, another note that is worth highlighting is lateral intraparietal cortex. I'm highlighting it not because I think that this is the place that the decision-making is done. I don't think there is any such place in the primate brain, and decision-making is I think decision-making is a distributed process that involves many nodes, but lateral intraparietal cortex has response properties that are also present in the other parts of the brain, and LIP provides a window into the dynamics and mechanisms of the decision-making process. Okay, so the standard work from Shadden and Newsom was that here the motion stimulus is presented outside of the response field of the LIP neurons, and one of the targets is placed in the response field of the neuron. So this is different. In the MT example that I showed you, the motion stimulus was in the receptive field of the MT cells, but here the target is in the response field of the LIP cells. And when the motion stimulus is presented, and while the animal is observing the motion stimulus to decide which saccade to make, we see that the responses in LIP show very interesting dynamics. When motion is strongly in favor of a saccade to the response field of the cell, the firing rates would go up. When motion is strongly in the opposite direction, the firing rate would go down. And for intermediate stimuli, there is a nice gradient of changes in firing rate with different slopes. Okay, so based on these observations, many cognitive and mathematical psychology models that have been previously proposed for decision-making have at least partially been validated. And those are models that suggest decision-making in a task like direction discrimination is done through integration of sensory evidence over time. And specifically, MT is thought to represent the moment-by-moment changes of the motion on the screen, and LIP representing the gradual integration of evidence over time. Now, these different PSDHs that are rising or declining at different slopes, shortly prior to the time that the monkey commits to a choice and makes a saccade, converge to a common firing rate, especially for saccades towards the response field. And this is also thought to be similar to accumulation of evidence to a bound or termination criterion for committing to a choice. There is a long list of papers that have found similar types of responses in different regions in parietal cortex, in different regions in frontal cortex, and also in subcortical regions, including striatum and superior colliculus. The psychological models that I had told you about are nicely captured by drift diffusion type of models, but there is also a long list of models that that have more or less similar properties, but different in exact quantification and parametrization of the model. So here the sensory input comes in, but of course it's a noisy input because of noise either in the sensory stimuli in the outside world or because of the neural representations in the brain that are noisy. And then the momentary evidence is integrated over time, and the decision-making process terminates when one of the decision bounds is reached. If the upper bound is reached in this case, it would, for example, be a rightward choice, and if the lower bound is reached, it would be a leftward choice. So models of this type quantitatively capture many aspects of the decision-making process. They explain psychometric function, chorometric function, confidence function, changes of mind, changes of the decision-making process and choices and reaction times through changes of reward, prior information, urgency. Many of different variations and flexibility, flexible aspects of the decision-making process are captured by these models nicely. Now, when it comes to hypotheses about implementation of this psychological model in the brain, a variety of models have been proposed, but in general, they make a few common assumptions. They usually assume that there are pools of neurons corresponding to making different actions, rightward and leftward, in the direction discrimination task that I showed you. And then these pools of neurons during the decision-making process would compete with each other based on the sensory information that would come in. And the competition is usually implemented through either lateral inhibition or feed-forward inhibition. Okay, so just to review, the common assumptions of models for implementation of the decision-making process is that there are pools of neurons dedicated to different actions and they compete with each other, and that the sensory information that would come in during decision formation biases the competitions between these action planning modules. But effectively, this means that if you engage the subject in a variety of sensory decisions, it's only the nature of the input into the integrator that would change, and the integrator itself would remain stable and fixed. Is that really true? Okay, so I told you that we chose two tasks to look at the interactions between the ventral stream for object processing and the parietal frontal or frontal parietal regions for decision making. One of the tasks that we chose was the standard motion discrimination task, but we also developed a new face categorization task that I hope would shed light on the validity of the assumptions of models that have been proposed for implementation of the decision-making process. Okay, so the motion discrimination task that we used is very similar to Shadlen and Newsom's. The monkey would fixate. Here, the shaded region indicates the response field of LIP cells. One of the targets would be presented in the response field, another target outside. Then the motion stimulus would be presented outside of the RF, centered on the screen, centered on the fixation point. And then there is a variable delay period. At the end of the delay, the fixation point turns off. That's the go cue for the monkey, and the monkey reports by making a psychedelic movement. The face task is very similar to the motion task, except that instead of showing the random dots stimulus, we show a stochastic face stimulus. The face is shown not exactly centered on the fixation point, slightly to the left or to the right. And it is because we wanted the different informative facial features, eye, nose, and mouth to be a more or less similar distance to the fixation point. Okay, so the stimulus in this specific example that I'm showing you is a continuum between a monkey face to a human face. There are morph stimuli in between, and the task of the monkey is to tell whether a particular face that is shown on a trial belongs to the monkey category or to the human category. The stimulus that we showed was also a stochastic. It was not fixed. Specifically, we were showing one frame of the stimulus followed by a mask, and another frame of the stimulus followed by a mask, and so on. On each frame, the features of the stimulus would change slightly according to the specific schedule that I'm going to tell you about in the next slide. But for now, just imagine that there are slight variations in the stimulus, and after each frame, we immediately mask it so that the perception of humans and hopefully monkeys that are engaged in this task is that effectively one face is shown and some dynamic cloud is coming and going over it. That is, changes that we make in the face are largely subliminal to the subject and not consciously perceived. Okay, so in addition to doing the monkey-human discrimination, monkeys also do an expression discrimination based on a sad, happy morph line. And here I'm calling these faces or these expressions sad and happy, not because I think monkeys perceive them as necessarily sad and happy, but just because it simplifies the communication of what we are showing to the monkeys. We know that monkeys perceive variations in the facial features, but what exactly their subjective emotion perception is, we don't know. Okay, so here is how we engineered our stimuli. Face stimuli are generally quite complex. They have many features based on the content of the face and also based on the spatial relation and spatial parametrization of the features in the face. And we wanted to simplify the stimuli to make them more manageable and provide better experimental control. So we chose 3 regions in the face, the eyes, the nose, and the mouth. And we morphed these regions separately in the stimuli that we generated. Anything outside of these regions, they are always set to halfway between the human and monkey face, or in general between the 2 prototypes, and therefore are not informative for the choice that the monkey is making. So by this manipulation, we have effectively made our stimulus a 3-dimensional stimulus. So there are 3 important features here, and that would make controlling the stimulus and making sense of the limited data that could be collected in a single session much more manageable. Okay, on different trials, we choose a separate combination of these 3 informative features. And from one moment to another moment, there are also small variations around a particular mean level for the morph level of these different informative features. Okay, so if the task is clear, let me quickly tell you about the behavior of the monkeys. For the motion discrimination task, the psychometric function, probability of correct as a function of the stimulus strength, goes from chance for 0% coherence stimuli to 100% for strongest stimuli. For the face task, there is also a very nice variation of the psychometric function from chance to perfect accuracy. The duration of the stimulus was also under experimental control, and for longer durations, the psychophysical threshold of the subject declined systematically in both tasks. Psychophysical threshold here is quantified as the stimulus level that corresponds to 81% accuracy, and for longer stimulus presentations, the effective stimulus strength that we need to achieve that level of accuracy is lower. That is, the subjects are taking advantage of the added time and extracting more information from the stimulus, which is compatible with integration of evidence over time in these tasks. Okay, the face task has a 3-dimensional input. The eye, the nose, and the mouth, they all fluctuate. We first ask whether a minor variation of the DDM can explain the behavior of the animals and also humans in this task. And the variation that we considered is of the following way. We imagine that these different features would come in and would be combined according to weights that the subject would assign to them, and weights that are specific for each context. After the linear combination of these features, we would effectively have a momentary evidence based on the face stimulus that is shown on that frame. And then these momentary pieces of evidence would be integrated over time towards a decision bound. So this part is pretty much similar to the DDM. Okay. We tested to make sure that the behavior and overall computational properties of the decision-making process in the face task are largely similar to the motion task. And I just summarized them for you. First, we looked at the time constant of the temporal integration of evidence to ask whether, similar to the motion task, for the face task, subjects have long time constants. The answer is yes, they have very long time constants in the order of tens of seconds, which means that there is minimal loss of information of the stimulus for the time period of the decision making in a trial, because the stimulus is presented for a few hundreds of milliseconds to a second. And if the time constant of integration is 25 seconds, for example, there would be minimal to no loss of information. We also checked if the weights that are assigned to individual features are stable over time or they are time varying. There are reasons to believe that they could be time varying, especially because responses of neurons in the sensory regions are known not to be fixed for a given stimulus, and they would have some form of temporal dynamics in which they initially rise and then they adapt and come back down. However, experimentally and based on the behavior of the animals, we now know that there is minimal to no variation of these weights over time. So there should be counter mechanisms that would compensate for the changes of the firing rates on the sensory side to keep the effective weight of the features fixed. So that mechanism, for example, could be normalization based on the overall firing rate of the neurons in the region. And finally, we tested if the integration across different spatial features happens prior to the temporal integration or after temporal integration. One can imagine integration of information from different regions of the stimulus over time and categorizing those features and then effectively using a voting mechanism based on the category of different features to render a choice. This early temporal integration would not be ideal because it would acquire less information from the stimulus. It would ignore interactions of the stimulus. However, it is in theory possible. We checked it, and we can also rule it out both in the human and monkey behavior based on the way that the psychometric function changes with time or the psychophysical threshold changes with time. Okay, so that's also similar to what happens in the motion task, where MT cells first represent the overall motion within a frame. So they do a spatial integration first, and then temporal integration is happening across frames. So for all intensive purposes, the computations that we can quantify for humans and monkeys in the stochastic face task is similar to the random dots motion task. So one last thing is that I told you the monkey does two different versions of the task: identity discrimination and expression discrimination. And effectively, we designed the stimulus such that the stimulus space is two-dimensional, not really the one-dimensional morphs that I showed you earlier. And in this two-dimensional space, a category boundary like this would separate different identities from each other, humans from monkeys. And a category boundary like this would separate different expressions from each other, happy and sad. Okay, so both humans and monkeys would take this contextual information very nicely into account. For the identification task and for the expression task, the behavior follows a very nice psychometric function. If I had superimposed the psychometric function for the context irrelevant dimension, for example, expression information during the face identity, we would get flat lines here. Unfortunately, I did not include that in my slide, but hopefully in future talks. And also, similar to what I showed you earlier, the integration of evidence over time seems to be occurring for both the expression task and for the identification task. However, the weights of different spatial features, different facial features, they are different in the two tasks. In the identity task, the eyes are the most informative features, although the nose and mouth also provide significant information for the subjects. And for the expression task, it's the mouth region that is most important or most influential for the decision making of the subjects. But again, the other two features also provide some information. Okay, so now responses of the LIP cells in the motion and the face tasks that I showed you earlier. For the motion task, we see a picture that is very close to the classic observation. Responses would go up or would go down based on motion towards or away from the response field. And there's a nice gradation for the strength of motion, and specifically when motion is weakly towards T-in, firing rates are lower, and when motion is more strongly towards T-in, firing rates are higher. For the face task, we see a reverse order. Now, for the T-in choices, when a face most strongly supports a saccade to the RF, the firing rates are lower, and when the face weakly supports a saccade to the RF, the firing rates are higher. So the firing rate order for the T-in choices is reversed. However, the firing rate order for the T-out choices remains the same as the motion task. What is going on here? Initially, when we had this observation, my reaction was that, well, something must be wrong. Like, we are really doing this task to replicate what we had in the motion task, and then look at the interactions between the IT and LIP. And I could not believe that we were effectively seeing a reverse order for the T-in. So we embarked on a long series of controlled experiments to make sure that we are not doing anything wrong. For example, many LIP cells have large response fields, and we thought that, well, maybe the stimulus is slightly overlapping with some of those RFs, and that could be the reason that things are distorted here. That is not the case. Pushing the stimulus to the opposite side of the screen and taking it very far from the RF would still preserve this order. We thought that well, there could be something wrong with the specific cells that we are recording from, but it doesn't really matter which part of the LIP you go to. This picture is repeated over and over. We modified the tasks, the size of the stimulus, it's like a bunch of different things. And no matter what we did, this reverse ordering of the PSDHs was preserved. Then at that point, we convinced ourselves that, well, this must be real. So let's look a little bit deeper into it, because from behavior and computational modeling, we know that more or less similar computations are happening in the two tasks. Why the responses of the cells, LIP cells, are so starkly different, qualitatively different from each other. Then we said, well, how about we look at the population responses instead of do the classic thing, which is averaging the responses of cells with similar saccade selectivity. Now, for the population analysis, I'm going to show you the simplest version that can be done, but I'm also happy to tell you about more elaborate analyses that we have done. So here, we can create a simple status space where different axes correspond to the firing rate of different neurons that we report from. And then we can do different types of dimensionality reductions, for example, principal component analysis, in order to create a lower dimensional visualization of the neural responses. So let's see how they look. In the face task, when we look at the pattern of activity at a particular moment in time during a stimulus viewing, that corresponds to different stimulus strengths, going from a strongly supporting T-out saccades to a strongly supporting T-in saccades, T-out and T-in with respect to the RF of the cell. We get an arrangement of the firing rate patterns in the population that can probably best explained as a curved representation in the status space. For the face task, for the motion task, we see a more or less similar thing, albeit a larger separation between the patterns that correspond to the rightward saccade and the leftward saccade, so T-in saccades and T-out saccades. Okay, so what's clear from these quote-unquote manifolds is that firing rates change in a very systematic fashion as the strength of the stimulus changes in this task. Also, they change in a very systematic fashion over time. Initially, about 200 milliseconds after the stimulus onset, the firing rate patterns that correspond to different stimulus strengths, they're kind of clustered together in the center of the status space and not very well differentiated from each other. But as time passes by, this clump of dots gradually expands, and the different stimulus strengths separate from each other very nicely in a way that both the strength of the stimulus and also the upcoming saccade are very nicely coded. So this gradual expansion of this pattern and gradual separation of the neighboring points is exactly the signature that you would expect to see at the population level for integration of evidence over time. So at least at the level of the population, there is a match between the two tasks. But what was going on when we looked at the average responses in the earlier slide? Here, effectively, what is happening is a rotation in the status space. So specifically, what we were doing was we're finding an axis in the status space that corresponds to the average firing rate of the population. So the weight for projection on this axis is 1, 1, 1, 1. When we do the principal component analysis, this vector in the high dimensional status space would also be a vector in the principal component space. And this is the vector of the average firing rate. And this is the manifold of the LIP activity for the face task. Now, when we project the firing rate patterns on the average firing rate axis, we see, because of the specific spatial relationship of the manifold, of the curved manifold, and the average vector of average firing rate, we see a reversal of the firing rates for the T in, but no such reversal for the T out, the stimuli. For the motion task, the manifold has a different orientation with respect to the vector of average firing rate. And then the projections on that vector would give us the classic ordering that previous studies have reported. Here, you may have noticed that these projection lines are not orthogonal. So is there a question? These projection lines are not orthogonal. In the high dimensional space, they are orthogonal. And because I'm just flattening everything to show you a two-dimensional picture, they of course would not remain orthogonal, but they are orthogonal. So don't worry about that. We are not making any mistake here. Okay, so what I effectively showed you is that there is a form of task-dependent encoding of the decision variable in LIP. The manifold at the population level that represents integration of evidence in the face task and in the motion task are rotated and shifted versions of each other in the status space. Put differently, we don't have just a single integrator or single attractor in the status space that reflects the integration of evidence. For different tasks, we have different attractors. Okay, so this difference would in fact have implications for the decision-making process. One of them is that the standard models that I showed you earlier for implementation, which were based on competition of action selective modules, that is probably not what is happening, or there are meaningful deviations from that classic picture. And the other thing is that the readout of the decision variable, which is necessary in the distributed computations that are happening in different nodes across the brain, that should probably be context dependent too. That is, the readout must be somehow context aware. So first, we wanted to make sure that the modification that we are seeing, or the differences that we are seeing between the face task and the motion task, are not simply because inputs are coming from, say, dorsal stream or the ventral stream. And for that, we decided to look at the two variations of the face task. So just to remind you, in the face task, we had a two-dimensional stimulus space and two different category boundaries. The stimulus could be chosen from anywhere in this space. And then once the stimulus is shown, depending on the specific context of the decision-making process, the subject would be reporting either the expression or the identity of the face. So here, the input, at least the physical input, remains fixed, but the decision changes in a context-dependent manner. So we can repeat our status space analysis, and here I'm showing you the manifolds for the integration for the species task and for the expression task, and again, they are not aligned with each other. So even when we keep the stimulus fixed and only change the rule that is applied to the stimulus for making a decision, there is a rotation and shift in the status space in the LIP population. And also this rotation and shift has important implications for readout, because if, for example, we choose the optimal readout for one of the task contexts, and then ask how well that would be that would perform if we project the other manifold on that optimized axis, we get biased decision variable readouts. So the decision variable that we optimize the vector for is going to be read out without any bias, but the other decision variable is going to be substantially biased. But of course, we know that the choices of the monkeys in the two contexts don't show such a bias. So that would mean that the readout of the decision variable from the population of LIP cells is also context dependent, and whoever that is reading them out is aware of the context and takes that into account. Okay. For the partial dissociation of the representation of the decision variable and action, I will show you just one piece of evidence which I think is reasonably conclusive. Here is the manifold that LIP population makes during a stimulus viewing. And here are the patterns of activity that corresponds to firing rates immediately prior to the saccade. And if the same population of neurons are representing both the action and doing the competition during decision formation, then you would expect that this and this would be largely aligned with each other, or in other words, this axis would effectively be this axis that I'm creating with my mouse, and the projection of the lines of the data points of the curved manifold would effectively land on different locations on this action axis in a very systematic manner. But that is not what we see. We see a nice separation between the action encoding axis and decision manifold in both the face task and also the motion task. In the motion task, that separation is significantly smaller, which I think there is about 10-degree angle in a three-dimensional space. But of course, if you go to high-dimensional space, the angle very quickly converges to 90 degrees, which would be expected. For the face task, given a three-dimensional projection, we have about 30-degree difference to 40-degree difference between them, which is quite substantial. Okay, so the approximation that has been made in the past modeling studies to think about decision-making as competition between action selective modules is reasonably valid for the motion task, although not totally accurate. But for the face task, it's not valid at all. And in fact, if we look at the selectivity of the cells for decision variable during a stimulus viewing and their selectivity for the final saccade, we see that about 25% of the cells have incongruent selectivity in the two periods, where 50% would be kind of chance match. And so here we have about 70% to 75% match. That would mean that there's a substantial difference in the way that cells encode these two different epochs. And more generally, I think it would make sense to think that we don't really want to rely on the average firing rate, and we want to take into account the context, and we also want to consider some form of separation between encoding of the decision variable and encoding of the action. And usually, the way that we think about it now is that there is some form of dynamics in which initially the cells in a frontal or a parietal area represent the decision variable, but then gradually, as the time for acting on that decision variable approaches, there is a transformation of the decision variable representation to the action representation. Okay, so I alluded to frontal nodes. A very similar type of responses that I mentioned are also observed in the frontal regions. For example, in the dots task, we recorded from the supplementary eye field and the pre-arcuate gyrus. And in both of those regions, there is a nice curved representation similar to what we saw in LIP in the face and motion tasks that I showed you earlier. And now we are kind of expanding our search, and it seems that the same patterns are repeated over and over, no matter where we look at. Okay, so we think that it's probably a brain-wide phenomenon and not something specific to LIP. Okay, so I'm going to spend maybe 1 or 2 minutes just to give you a little bit of intuition about what we think is happening for the emergence of the curvature. And upfront, I think we can explain one aspect of the observations that we've had so far, and that is the presence of the curvature, but we are not able at this moment to explain another aspect of it, which is why it is context dependent. Okay, so why curvature should exist at all? I think there are good reasons to support that based on efficient coding. Imagine that you want to represent integration of evidence in a population, a status space. The standard way to think about it is line attractor dynamics, or mechanisms that approximate line attractor dynamics. And in fact, all the earlier models that I told you would fall in this category. And effectively what they make is a form of line in the status space with a nice gradation based on the stimulus strength. So you would expect to get something similar to a curved manifold, a linear manifold. But of course, we know that firing rates in the brain are limited. You can't have negative firing rates, and the high firing rates are probably also not encouraged because of the metabolic cost that they have. And if we take into account the boundaries of firing rates that must exist in a realistic neural network, we effectively say that the space that is usable is, for example, like this cube. And if you want to fit a line in a cube, well, you can always make it smaller. But if you make it smaller, then the neighboring points, different stimulus strength, the patterns of activity that correspond to them would be more similar to each other or closer to each other, and therefore probably less distinct from each other. It would make a lot of sense to create some form of curvature to increase the separation of the neighboring points from each other, and also avoid the center of the cube where the majority of the cells in the population would be firing at a relatively high level. So curving the manifold would provide those advantages, but of course we don't want to have too much of twisting in the status space, because that would also complicate the readout for us. So let's scratch that. We want some form of simple nonlinearity that would help efficiency of the coding and also would allow a simple type of readout. So reading out along a quadratic curve or a linear projection of that is a relatively straightforward thing that could be easily implemented in various models. And we collaborated with Christian Markens and Alan Manku to ask whether a simple RNN that is trained based on the efficiency principles that I told you earlier would in fact replicate the curvature. And the answer is yes. So after we train the RNN and we repeat our analysis on it, we effectively also see a curved representation. But the thing that I told you we cannot explain at this moment is why, as a result of the context or the rule of the representation, the curvature or the manifold changes in a status space. I have ideas about it. I'm more than happy to share them with you, but they are speculations at this moment. Okay, so overall summary. I think a weighted linear integration of evidence over a space and time nicely explains fine face discrimination and presumably a large number of other types of object discriminations. The neural mechanisms, I think context should be taken into account, readouts should be context aware, and also some form of separation between encoding of the decision variable and action should be taken into account. At this moment, we're also looking into responses of the phase patches in intratemporal cortex, and hopefully in the coming months or year, I can share more information about that. There is plenty to learn from them as well. And at the end, I would like to acknowledge our collaborators, specifically Goki, who led this project with extreme brilliance and competence, and he deserves really all the credit. And Christina and Christian and Alan, who helped us with various aspects of the project. And finally, our funding sources. Thank you so much.